From Microbe TV, this is Office Hours for Wednesday, July 31st, 2024. I'm Vincent Racaniello, and welcome to your special corner of the viral internet, where you can ask questions about viruses and so many other things. Coming to you from New Jersey tonight, where it is 29°C and cloudy. A lot of humidity out there. It's just sitting out there for a few minutes. 69% humidity. Welcome, everybody. Tell us where you're from in the next few minutes. I have a few things to do first. First, I want to thank our moderators who are always keeping this civilized. Barb Mac, UK, Les from California, Steph from San Francisco, Vanity Nutrition from Long Island are with us so far. And yes, I got a haircut, and I also have black glasses tonight. I decided to go with the black, I'm wearing black clothes. The other day, yesterday, in fact, I was walking up 7th Avenue. I was wearing black shoes, black boots, a gray pair of pants, a black turtleneck, a black jacket. And it's really hot, and I have a jacket on because all the places I go to, air conditioning is too cold. And there was a guy sitting on the bus stop, and as people went by, he would say, "You're not from here. You're not from here. You're not from here." And when I passed, he said, "You're not. You might be from here." Because who the hell else is walking up 7th Avenue on a sunny New York day with black? Anyway, black glasses, black shirt, black pants, which you can't see here. I have a few things here. Let's see, to do. Tell us where you're from. By the way, you notice that the image for this has a medal around my neck. Of course, I didn't earn that medal. It's an Olympic gold medal. It's a picture of it that I stuck on here. But I wanted to remind everyone that that is a icosahedron, right? An icosahedron. What is an icosahedron? Well, here it is. Here's the medals, right? Gold, silver, bronze. And they have this inset, which is basically a hexagon, right? But in virology, a hexagon is a surrogate for a kind of structure of particles. So let me actually put that up here. I have the lecture queued up for later, but I have the first slide which tells you all about icosahedral symmetry. Now, what is this? I don't think we ever did structure in this as a mini lecture. Right? We'll have to do it another time. So this is an icosahedron, right? So it just looks like a hexagon. If you took all the stuff in the middle off, it'd be a hexagon, which is what's on those medals. Let's put some of Vinny up here. So at the bottom here is how icosahedral capsids are built, right? It is a three-dimensional structure, of course, but in one plane, if you look right at it, it is a hexagon, which is so. That's why these Paris gold medals, which where is it? Get rid of the screen share. There we go. The little hexagon in the middle. You know, if you filled in a few lines, there would be viruses. Every time I see them, I think of viruses, and I hope you did too. Tell everyone those are viruses on those medals. And they'll go, "What?" Anyway, I thought you should know about that. I think it's pretty cool. All right. So much for that. Paris viral medals, right? Let's see where everyone's from. So Chris Lipowski is in Montreal, Canada. A lot of rain, thunderstorms up there. Abdulaziz is from Saudi Arabia, which is where it's 30°C. Lise is in Columbus, where it's 84 in thunderstorms. Rima, it is baking heat in Iowa. Wayne is from Urbana, Illinois, 82F, 28C. Nebby is in Central Florida. MN is outside D.C. Yes, give this a thumbs up. Right now, go below the video window and there's a thumbs up button. Just hit it. Maybe we can get more people here to check this out. Karen's playlist is in northern Maine, 74F rain. I am back at the incubator. I've been there for 2 weeks now, and I've been doing some recordings there. But this one is at night. It's late, and sometimes I don't feel like staying at the. I have stayed at the incubator, earlier this summer, and then it's 10 p.m. and I have to get home. I don't feel like doing that right now, but I'll resume being at the incubator. But today I was at the incubator. Yeah, and I took an early train home. Brian is in Montreal, 24C and Sticky. Fire EMS is in Atlanta. Rima says they're crazy winds, 126 miles per hour in Iowa. By the way, early on, I think it was Barb Mack said, "Oh, Vincent has a gold medal." Of course, I don't. I don't have a gold medal. These are just, where is it? These are just the medal that I photoshopped onto here, right? But I did get a gold medal once in my life, and Vanity Les points it out here. I received the Richard Ernst medal. I went to Zurich to give a talk. It was a lovely ceremony. You can go to that link and see the whole talk. And I got a gold medal. Richard Ernst was one of 3 people who did the work to allow the MRI to be developed, and they got Nobel Prizes for that. Each of the 3 got Nobel Prizes. And so Ernst started a lecture series to reward scientists who communicate with the public, because he thought that was really important. He learned that from making the MRI, where, of course, this is a direct patient application, right? And so you have a lot of interfacing with the public, but he thought it was generally more important. So I got that award at the end of 2019. Actually, they awarded it, and the committee met and decided in 2019 before the pandemic. And then I don't remember, 2022, I actually got it and I went over there. It was really cool. So it was a gold medal. I got one, but not an Olympic gold medal. Okay, Kang is from Chicago, 29C. I know John is from Minneapolis. Steph is from San Francisco. Yes, here we go. Barb Mack. Vincent's wearing a gold medal. So that I do have a gold medal, but not that one. I didn't perform in any Olympics. Carol, our favorite nephrologist from Bakersfield, California. Jessica is from Toledo, Ohio. Yes, I got a haircut this weekend. It'd been like 2 months since I got a haircut. And it is a little too neat. It looks like too beanie stuff here going on, right? But it does make me look younger, I think, right? By the way, I don't dye my hair, okay? People always ask me. No, I don't dye it. I never have. There's some colorless hairs up here, but you can see my beard is white. That's fine. But this is not dyed. Fields of Green is from Salinas, California. Yeah, Rima's in Iowa, and John is in Minneapolis, 72F. Yeah, Jessica's representing the University of Toledo. Carol in Bakersfield is 101F. Lucy from Sechelt, BC. Jonathan's from Encinitas, California. Mark is from Edmonton, Canada. Joseph is from Ontario. Abdul Rahman is in Brooklyn, driving to Buffalo in 20 minutes. Oh, that's quite a drive. It's way up there. Bernard is in Salisbury, Maryland, 78. Humidity 84C. Peyton is in St. Louis. Joseph is in Austin, Texas. Maureen is in Northeast Ohio. Pat is in San Fernando Valley, where it's 97. Veronika, privyet. My Russian is increasing. If I say to you, "Как дела," you would say, "Хорошо," something like that. But anyway, we're not having a conversation. So it's privyet. In Moscow, Pleasant 16C. Well, that's cool. I wanted to write some comments about the live stream with Professor Schmidt. Go ahead. Yeah, that'd be cool. Well, HIV is not icosahedral. It's enveloped. Okay, so it wouldn't have that form on the metal. Cheryl is from Santa Rosa, California. It reminds you of Orgo. Yeah, hexagons, right? That's popular in Orgo. But it doesn't remind me of Orgo. It reminds me of viruses. Frank is from Santa Barbara. Les says the shape of viruses is related to number theory. Yeah, well, icosahedra have very specific structures, right? It is it's a three-dimensional solid with each face made up of equilateral triangles, a certain number of equilateral triangles. Lise thinks of a benzene ring. Yeah, organic orgo. Most people didn't take virology as undergrad, so the first thing most people do take orgo, so that's what you would think of. Yeah. John says there's more math and geometry than I expected in the virology course, but I don't know why that surprised me. Yeah, there's a lot of regularity in the virus world. Let me go back to this this slide. So it is made up of equilateral triangles. There are 12 of them that make up 60. 60 equilateral triangles make up each of the five fold axes has 5, and then there are 12 making up 60. But the actual viruses are spherical. They're not icosahedral. Okay, it's just that they're arranged by icosahedral symmetry, and we'll talk about that. I'll do a mini lecture on structure if I haven't done it. I'll check, but we'll do a mini lecture on structure. That's very cool. Tracy's in Chapel Hill, where it's 27. Patricia's in D.C. Lori and Mike are in Seattle. Lane is in southern Alabama. Mug 1 is in Pueblo, Colorado, 98F. Martha's in Charleston, South Carolina. Questella's in Ohio. Lori is in Charlottesville, Virginia. Les says 101 puts the F in Fahrenheit. We all know what that means, right? Wayne says Gons are 2D and Hedra are 3D. I suppose that's correct, yeah. Rainey is from eastern North Carolina. Yeah, this is one I've not seen before. Professor Special K from central Illinois. Margot is in Phoenix, where it's 102. That seems to be the highest temperature tonight. Okay. Tomorrow, Thursday, August 1st, we will publish the 50th episode of Matters Microbial by Mark Martin. And Mark is having a giveaway. He's giving away stuff, so you can get a sticker, and a pin. I have to pull this back to get in my focal plane. This is a sticker, episode 50. This is a pin that says episode 50. These are free if you want. Just send an email to rsvp@microbe.tv. Why don't we put that up here? rsvp No, oh, I can't spell. RSVP at microbe.tv. It doesn't need to be so big. I think we can do 48. Okay, you send an email to that with 50 in the subject line, so we know it's about the 50th episode of Matters Microbial. You'll get a pin and a sticker. By the way, the pin has a plastic cover on you have to take off. You know, some people get their Apple chargers and they leave the plastic on them forever because they don't know it's there. But it's to come off this. So free stuff from Mark Martin, courtesy of Mark Martin. He's very excited about his 50th episode, right? He does one a week. It's pretty cool. So show your love for Matters Microbial. I think we have Nebby now from Central Florida who has joined us. Did I already do that? Yeah, I already did that. Sorry. Here we go. Betty is from Washington Heights, New York City, my old stomping ground. That's right. Spent many years there. 38 years, pretty much, working up in Washington Heights. Ian is from Dunedin, New Zealand. Kathleen is at the Jersey Shore. Visto is in Sydney, Australia, where it's 18C. Theodore, where's Theodore from? I don't remember from last time. From Greece, maybe? Kiakidis or Kiakidis, I don't know. Sounds Greek. Okay, that'll do it for Who Are We? If I see some new ones, I'll certainly answer them. Let's see, anything else? All right, I have a mini lecture for you. Let me answer a few questions first. And Les says, with TWIV and Immune, I don't just keep up with the fields, but I listen in on how scientists think. That's right. We read the papers and we try and understand what works and what doesn't work, what experiments we would do, so you can hear how scientists think. I think it's valuable. There's a subset of people who don't care, right? I don't care if you don't care. Your life is good because of science. And you can deny science as much as you want, but hey, it's working. Let's do a few questions here. I know I said that already. Rima wants to know what viruses maggots carry with them. Insect viruses, right? There are lots of them because they're larval forms of flies, typically. Flies have viruses, and so do the maggots. Same viruses, most likely. Christopher in Montreal wants to know, why are cases of COVID increasing this summer? So this is a good question. We talk about this with Daniel all the time, right, on the clinical update, which is going to be recorded tomorrow, by the way. Now let's take influenza, right? Influenza is a winter disease predominantly in temperate climates. So, but also it's seasonal in other climates. But in a temperate climate, it's a winter disease. There's a little bit of flu during the summer, but the curves go way down, and you don't see those few cases throughout the summer months there. But COVID now has a big peak in the last month or so. And it's slightly different from the peak last year. So it's clearly, COVID is clearly a winter respiratory disease because it occurs in the winter. Every winter there's a surge, right? That is in climates that have winters, which are temperate climates. But it also says surges in other parts of the year. And now there's one in the summer. So why is that? Well, first, obviously, what we think about for flu is that the weather conditions and the social conditions in the winter promote transmission. So people are all in rooms and houses and whatever because it's cold out, so they're not hanging out outside. So they're inside, and it's low humidity and it's dry. Well, that's low humidity, sorry. It's low humidity and cold, which promotes the transmission of the droplets, the respiratory droplets that contain influenza viruses. Now, you would think the same thing would happen with SARS-CoV-2, but it doesn't have to. It's a different virus. It's structurally very different, even though it's enveloped. It's different, totally different. And so it may not have the same requirements for low humidity and low temperature. And so it may be perfectly happy transmitting in the summer. And the summer is not a bad time to transmit. People do get together, right? College students move around. They go from college to home and they carry their viruses with them. People do things indoors a lot during the summer. Sporting events still happen during the summer, right? Look at the Olympics in Paris. Everybody's sitting inside next to each other. Great situation for spreading a respiratory infection. I don't see many people wearing masks, so they're not concerned about it. I mean, it is up to them, right? I'm sure if some of them were immunocompromised in some way, they would wear a mask, and that's totally fine. So that's the story. I don't know why. Well, SARS-CoV-2 is a different virus, so it's going to have no reason to expect it's the same as influenza. Even at the beginning of the pandemic, I will say, I and maybe others said, yeah, that will probably settle into a winter respiratory disease pattern. But that was ill-advised, I think. And now it seems like it's just going to occur whenever people are moving around, right? Yeah, so currently there are a lot of cases. It will probably go down in the fall. Or maybe it will sustain through September, late August, September, when students go back to school. And then it will go down, and then there'll be a winter peak. December, January, February, it will go down. And then maybe a spring peak when students move around as well. I mean, we really need a few seasons worth of experience to try and understand what's going on. So that's a good question, Christopher. Those are my thoughts on it. Steph knows a few people with the common cold. Common colds also transmit during the summer. They're predominantly winter, right? So the big peaks are in the winter, but they still occur. I mean, even influenza occurs in the summer, but there are very few cases. And this is the same with common colds caused by any of a number of different viruses, rhinoviruses, paramyxoviruses, bocaviruses, adenoviruses, and so forth. Chris wants to know, how can we best protect our high-risk family members who don't travel from those of us who do travel and bring viruses home? Well, if you're living in the same house, then so what you can do is wear a mask when you travel. And that way you're reducing your chance of bringing anything home. Now, it's not just COVID, SARS-CoV-2 that you're worried about, right? You're worried about respiratory syncytial virus, influenza viruses, and a number of other respiratory viruses. So in my opinion, wearing a mask whenever you're in a crowded area, certainly on the airplane, but if you're at a meeting, wear a mask, and that way you're less likely to bring it home. And then you might consider wearing it for a period of time at home. I mean, the best thing would be if everyone did, but that's weird in the house, right? It's up to you. The simplest is for the traveler to wear a mask for, say, a week to get past the major shedding period for most of these viruses. And I agree. Excuse me. Sorry. I agree this takes sacrifice. You don't want to wear a mask at home, right? But if you want to protect people who are at risk, that's what you have to do. Okay. Kang wants to know, do the viruses that are spread by vectors and infect and cause disease in human hosts also infect and cause disease in the vectors? Are these viruses benign passengers in the vectors? So by vectors, I presume you mean like insect vectors, mosquitoes and ticks, flies of certain types, right? So as far as I know, they don't cause disease in these vectors. So they are benign passengers. Now, so that's a vector. A mammal, say a mammalian host other than humans, it wouldn't be a vector, that would be a reservoir. and they can be sick depending on what animal it is. But the mosquitoes and the flies and the ticks, no, they don't get sick. Well, they don't die. You can infect mosquitoes in the lab, and they remain viable. Okay, so Veronica wanted to make a few comments about Michael Schmidt's live stream. So let's go through those, and then I'll do the mini lecture. So Veronica said, it's not exactly true that in the USSR antibiotics were hard to come by. In the country, they were freely sold in drugstores. Many people, including my granddad, developed the habit of using antibiotics whenever he got sick. Didn't matter that a cold isn't caused by bacteria. I also found out that when my grandma got dysentery in the late 30s, she was treated with phages. Wow, cool. Also, with respect to botulism, there was a huge outbreak in Russia this month. 417 people got sick, 2 died. The cause, badly made beans, which were then used by many firms that deliver pre-made food. Ooh. Some hospitalized people made videos of their neurological damage. It looks really scary. That's really unfortunate. I'll make sure to let Michael Schmidt know about that. Tell him that there were plentiful antibiotics. Many countries today, antibiotics are available over the counter. You don't need a script, and they are abused in other countries, as you say. They were abused in your country. Yeah. All right. Let's do a mini lecture, about 30 minutes or so. Then we'll come back to questions. So it's 8:30 local time here, and I want to do a topic that is relevant to all viruses, not just to one or a few types. All right. So first, I thought I learned last time how I could do full screen. Let's see if I can do that and do it right. But first, I have to put on me. And then, do I do full screen? Try with that. Let's see. I don't have my pointer now. Where is it? It disappeared. Darn. I have to do the settings. Show. No, show pointer when using the mouse or trackpad. It was on the wrong setting. I have to see in my replay here. It's not too much delayed tonight. I'm just seeing myself make the change in setting. Yeah, it's working. Cool. So this is called the infectious cycle. It's the second lecture of the virology course. And it's all about what goes on in an infected cell and how we can measure it. So an infectious cycle is the name we call all the events that happen inside of a cell. Between the time the virus gets in, the virus particle gets in, makes all the molecules needed to make new virus particles, and then gets out. So that whole thing is called the infectious cycle. And we divide it into steps, Well, we have to study it, basically. But it's a continuum, of course, like attachment and entry of the genome, translation if it's an RNA, replication, protein synthesis, assembly. Now, for many viruses, they're DNA viruses. This is an RNA virus, poliovirus. DNA viruses get in the nucleus. The DNA has to be made into mRNA, which will then come out into the cytosol and make proteins and so forth. So that's the infectious cycle. And before we go on, we have to make some definitions. These are words that I use very specifically, but doesn't mean the same thing to a non-virologist. So a susceptible cell has a receptor for the virus. That's all it means. It means the virus can attach and get in, but it doesn't mean that the cell is going to support replication. It may or may not. So this cell on the top right is susceptible, but it is not able to support replication of the virus. So no new infectious particles are made. But so susceptible solely tells you, when we're talking about a cell now, that there's a receptor on the cell for a virus. Now, a susceptible host is a different story. We're just talking about cells in culture here. A resistant cell has no receptor. So here, second row, these two cells have no receptors. They're both resistant. Now, it turns out that the cell on the left can actually support a viral replication cycle, but it doesn't matter because the virus can't get in. It has no receptor, right? A permissive cell has the capacity to replicate the virus. It may or may not be susceptible. Permissivity refers to the internal environment of a cell, whether all the machinery is present that is needed to make new virus particles. Permissivity. So on the left, we have a permissive cell, which is not susceptible, has no receptor. So this cell will not support replication because the virus can't get in. But the one on the right is susceptible and permissive, and that's the only cell that can take up a virus and replicate it. Okay, so those are the definitions. Now, in the early days of virology, remember viruses were discovered around the end of the 1800s, early 1900s. We couldn't grow them in cells in culture, so we used animals and plants to propagate viruses. But eventually we learned how to do cell culture, and today we do that. We have cells growing on plastic surfaces in the laboratory covered with liquid. I think we have a picture of that in the next slide. And there are different kinds of cell cultures. There are primary cells where you take a tissue and chop it up, separate the cells, put them on plastic, and they will grow and make a nice, what we call monolayer. These are foreskin fibroblasts. Why, you may ask, foreskins? Because the hospitals get lots of them every day. Babies are born, clip, goes the foreskin, and they throw it out, but you could get it. You could get a human research protocol approved in your institution, and you could go in, and when they give birth, you could get the foreskin, you could take it back to your lab, cut it up. I could not do that. I don't think I could cut up a foreskin. I'm sorry. Chop it up with scissors into tiny pieces and mix it with trypsin. You get individual cells. Oh, like Woody Allen said, don't mention scalpel or knife and penis in the same sentence, right? It's cutting up a foreskin. Then you put them in culture, and you can grow a lot of viruses in them. But they don't grow forever. They divide a few 20, 30 times, and then they die. But we have cell lines that will grow forever. There are mouse cell lines. Then, of course, HeLa cells, the famous HeLa cells from Henrietta Lacks. They're immortal, but they're also not normal. They're transformed. And you know what that means because we had a little mini lecture on transformation. So here's this is the slide I wanted to show you. Yeah, I know my image is covering the right side, but I don't think it matters for most of them. I'm looking at it here. If it does, I'll fix it. So this is a picture of my incubator in the lab, which doesn't exist anymore, but when it was running. And these are various kinds of plastic containers where we're growing cells. And the simplest ones are these dishes. These are 6 centimeter and 10 centimeter dishes. They're plastic. The bottom is covered with cells, and then we have a little bit of medium in them for nourishment. There are also flasks and 6-well plates, as you can see, different sizes of flasks, right? The one on the bottom and the ones on the top are bigger. And then this whole incubator has 5% CO2, which buffers the medium and keeps the pH right. You can also grow cells in these spinner flasks, where if you need a lot of cells, billions and billions, then you grow them in suspension in liquid. They don't stick to the plastic because the area of the plastic is limited. But this is a liter. You can get 100,000, no more, 400,000 cells per ml. And so you can get a lot of cells. Okay, so that's how you grow them in the lab. And I just want to remind you that HeLa cells were derived from Henrietta Lacks, and her story is told by Rebecca Skloot in the Immortal Life of Henrietta Lacks. HeLa, of course, is the first 2 letters of her first and last name. And if you've read it, you know that I am mentioned a few times along with TWIV, which Rebecca liked very much. And we talked about, she contacted me early in the development of the book, and we stayed in touch until after it was published. And she got too famous and now doesn't talk to me anymore. Now, when you infect cells, often the cells change. Often they die, but not always. And we call the changes in the cells cytopathic effects. And so here's a monolayer of cells that we're going to infect with poliovirus. But on the top left, these are uninfected cells. You see, they're nice. A monolayer means there's one cell high, and they're all touching each other. They're all sitting there nicely behaved, beautiful cells. This is a gorgeous cell culture. And then you infect this with poliovirus, and at 4 hours after infection, you can see the cells are already changing. Many of them are rounding. And then at 8 hours, most of the cells have come off the plastic. They're floating in the medium, right? You can see them here all rounded up and floating. And then at 12 hours, all the cells are dead. Most of them are broken, as you can see on the lower right. So that's a form of cytopathic effect. And there are actually other kinds as well, but it's a way you can tell that your cells are infected. And I always like to show this paper to the students to show the use of cytopathic effect. This is a paper published in January of 2020. This is in Nature. A pneumonia outbreak associated with a new coronavirus of probable bat origin. This is from the lab of Zhongli Shi in Wuhan, the Wuhan Institute of Virology, right? So they picked up this virus early on, and they isolated it from a patient. And here in this paper, they describe it. And here I've highlighted, here we report the identification and characterization of a new coronavirus. The old name was 2019 nCoV, new coronavirus, but it was then changed to SARS-CoV-2, which caused an epidemic of acute respiratory syndrome in humans in Wuhan, China. So what do? So they isolate the virus. What they do is they take a bronchoalveolar lavage fluid sample. They put a tube in the chest into the trachea of the patient. They put a little PBS in, and then they pull it out. So that's a lavage. You're washing the trachea with PBS to try and get something out. So they get BALF from one patient, an ICU patient number 6. So this is a very sick patient. They take this virus and they put it on cells in culture, 2 different kinds of cells in culture. And after 3 days, they get cytopathic effects. See, on the left, we have the uninfected cells. And on the right, we have 3 days later, you can see cytopathic effects. The cells are dying and detaching, just like I showed you for poliovirus. I wonder if any reporter ever bothered to look at this paper. This is so cool that you can do this. They take fluid from a patient, and they put it on cells. Can you imagine 3 days later, the person who comes in and looks at this under the microscope, and they say, "Eureka, it's a virus," because this is typical of viruses. Well, anyway, we'll come back to these cells in a bit. Another kind of cytopathic effect is fusion of cells. We call that formation of syncytia. So here, an infected cell will fuse with another cell and make a cell with 2 nuclei, and then eventually more confused. You get big cells with many nuclei. Those are called syncytia. And I'm laughing because Brian Silverman asked the question, which we'll get to in a moment. And here is an actual cell culture with individual cells and then a syncytia here. You can see a fused cell. Let me go back. Remember, I called this the incubator. Yeah, it's a cell culture incubator. That's why we called our studio, our recording studio, the incubator, because we use incubators in the lab to incubate experiments, right? And incubator also has a connotation of being a place where things are developed. So it was a perfect name, right? So Brian asked, the incubator in the lab, will you at some point have a lab in the incubator? No, we won't. You can imagine we might make a little lab and show how to do experiments, but there's just too much regulatory issue associated with having a lab, and I don't want to do that. I'd borrow a lab. I could go to find someone's lab and film in it or record in it if we wanted to do that. All right. So syncytia is another form of cytopathic effect. Let me talk a little bit about how you count the number of viruses in a sample. This, by the way, is Amy's bench, lab bench, when she was in the lab years ago. These tubes all have virus in them. Here's some plaque assays you can see with the purple staining here. And, well, these are labeled. They're kind of falling all over the place. But, she knew what was what. They're all labeled. So we measure each of these has virus in it, but we don't know how much. We can measure it by infectivity, or we could count virus particles and their components. I'm going to talk a little bit about how we do that. Of course, the most famous assay, the plaque assay, which I think is the most incredible assay ever devised, is an assay where you make dilutions of virus. What is a dilution? Not a delusion, a dilution. You take a virus. I give you a tube of virus. I say, how much virus is in here? It's poliovirus. And you take 0.1 ml and you mix it with buffer, 0.9 ml of buffer. And that's a 1 to 10 dilution, or 10 to the minus 1. And if you keep doing that, you take 0.1 ml of that and dilute it again. You get 10-fold what we call serial dilutions. And you can see you can go up to 10 to the minus 7. And then you take a little bit of each of these 0.1 ml and you add them to cells in culture. You take 3 dishes here and you remove the medium. You add the virus. Well, you add this dilution. You don't know if there's virus in it yet. You let the virus absorb for 45 minutes. Then you add an agar overlay and put them in the incubator. And plaques will develop, these clearings in the monolayer, which are caused by individual viruses infecting one cell and then many, many more. And I'll tell you how that works in a moment. But you can count these plaques and figure out the titer of the virus in plaque-forming units per mil. So one virus will form a plaque. We call that a PFU. So here there are 17 plaques in the middle plate. And so the dilution here was 10 to the minus 6 and another 10-fold. So that's 10 to the minus 7. So the titer would be 17 times 10 to the 7th, or 1.7 times 10 to the 8th. Just move the decimal point over and we add another number to the exponent, okay? That is that is the way you would figure out the number of infectious viruses in a sample. So what is a plaque? On the top here, we have we're expanding a very small area of the plate, and you can see individual cells. And so where this hole is in the middle, this single cell has been infected with a virus, and the virus is released, and then it infects surrounding cells and kills them, and then this hole gets bigger and bigger. And when you stain the plates, which is what we did in the previous slide, you can see the hole very clearly. And you have an agar overlay on to restrict the diffusion of the virus. If you had a liquid overlay, of course, the virus is released from this first cell. It would simply be infecting all the cells and killing them all. Hold on, I have to send a text here. So Les, I'm sending you a text. Please take a look at it and act accordingly. This on the bottom is our other views of a plaque. Here's a single plaque under the microscope. You can see there are dead cells in the middle. And so this will eventually be a hole that you'll be able to count. And some viruses don't form plaques. And so we can put a gene in the virus for a dye that gives it a color, and then we could count plaques as well. So the plaque assay is beautiful. Now here's a movie of plaque formation. Let me stop this and tell you how it was done. So if you remember last week, I showed a wooden plaque made for me by Jeff Smith over in the UK that says TWIV on it. It's a piece of wood. His lab did this experiment. So there's a monolayer of cells they infected with vaccinia virus. And they looked under the microscope and they found the beginnings of a plaque, a little hole in the monolayer. And then they took a frame every so many minutes and did time-lapse photography. So now you can see the plaque develop over like 15 hours. So here we go. You can see it gets bigger and bigger. The cells around the edge are dying and then they detach. Isn't that amazing? And then, of course, when you take off the agar overlay, all those dead cells come off, and then you put the dye on and you get a hole in the monolayer. I think this is the greatest video ever made. Anyway, that's how a plaque works. And in my office, I used to have this, what I call the wall of polio, right? These are 6 well plates, each one with a plaque assay done in them, 1,600 and some 6 well plates. And you can see the dye, the staining, the purple dye that we used to stain the plaques. You could still see it faintly here, right? And I had a postdoc years ago who did an experiment, and she used 1,670 plates. And so I said, don't throw them away. And I built this wall in my office, and people used to come. So these are some of my virology students of a few years ago who came and said, let's get our picture in front of the wall of polio. So they had just come for office hours, right? And I loved to do this. I thought it was so cool that people came and wanted to have their picture in front of this. Now, these are plaques, actually, of influenza virus. We also did that in the lab a little bit. How many viruses do you need to make a plaque? Do you need one or do you need more? Well, the answer comes very easily. You do an experiment where okay, so Artemis wants to know, what are the bright spots? So when the cells die, they become refractory to light. So the light passes through the other cells, but these now become refractory when they die, and so they become bright. So all the cells here that are becoming white are actually dying. Okay, how many cells does it take to form a plaque? So what you do is what we call a dose response curve. More math, John, okay? It's very simple. You make dilutions of virus on the x-axis, and you do a plaque assay with each dilution, and then you count the number of plaques. And if one virus is enough to form a plaque, you're going to get a straight line. That's called one-hit kinetics. And for most viruses, one virus is enough to make a plaque. For some viruses, you need more than one. For some, you need 2 or 3 or even more. For 2-hit kinetics, you get this parabolic curve here. All right, so for the animal viruses that we have worked on, it's one-hit kinetics. So that's how we know one plaque is started by one virus. And so you could pick a plaque. You could pull the agar plug above a plaque, put it in some buffer, and that's called plaque purification. That's how we make virus stocks. We do it a few times to ensure that we're getting the progeny of just one virus. Now, some viruses don't make plaques. And so for them, we do endpoint dilutions. If you've ever seen these phrases called TCID 50, tissue culture infectious dose 50%. Well, that's the amount of virus that would infect 50% of cells in your culture. And what you do is you take a multi-well plate. So you have wells across and rows down, and you infect each row with a different dilution of your virus, -2, -3, -4. And then you incubate and you just look for cell killing. And so then you score the cell killing or cytopathic effect. You look for the one with 50% killing. And so there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 wells here. And so 1, 2, 3, 4, 5. The -5 gives us 50%. So the TCID 50 of this virus would be 10 to the 5th. 100,000 viruses, plaque assay most likely, well, it's a TCID 50. 100,000 TCID 50s are enough to infect half of the culture. Okay, the other way we study viruses is by what we call a one-step growth cycle. And this was first done in the 1930s to study phage reproduction in E. coli. You would let the virus stick to the cells. You dilute the culture to stop any more attachment. The virus would start to replicate. Then you'd sample and assay by plaque assay. And what the virologists found, to their surprise, this is in the late 30s now. When you infect cells and you measure virus production here on the y-axis over time, there's a period where you don't see anything. They were so surprised because with bacteria, it's totally different, as I'll show you in a minute. There's this eclipse period where there's no virus produced. And then after a certain period of time, you get virus production. And this showed them for the first and definitive time that viruses were not small bacteria, because many people thought they were up until this point. So an eclipse period is a defining feature of viruses. Now, why? Because bacteria don't have an eclipse period. When you put a bacterium in broth, they pretty much start to divide right away. 1 goes to 2 to 4 to 8, 16, 32, etc. So this is the bacteria divide by fission. Viruses don't do that. The virologists said, oh my gosh, there's a period of nothing, and bacteria don't do that. So these are a unique thing. 1939, that was the eureka moment. We can do this with eukaryotic viruses too. Here's a growth curve for adenovirus. Again, we have an eclipse period after infection where we can't detect any infectivity, and then we have virus production. You can see the time course for adenovirus is long, 24, 28 hours before all virus production is done. But this is a very powerful way of studying viruses. And finally, let's end up with a few thoughts on measuring virus particles. So we know that we can measure infectivity. I've just shown you that. But not all viruses will plaque, and sometimes it's hard to do infectious assays. Like SARS-CoV-2, you need a BSL-3. So what else can we do? Well, you can do a few things that are listed here. You can do hemagglutination. You can use red blood cells. I'll show you what that is. You could do electron microscopy, but it's not done that often. You can measure enzymes in virus particles. You can use antibodies. You could do immunology. You can detect viral antigens by using antibodies. And of course, you could sequence the genome. You could look at the nucleic acids. So let's just look at a few of these in detail. Hemagglutination relies on the fact that for some viruses like influenza virus, the red blood cell has a receptor. Now, the virus does not reproduce in red blood cell, but it will attach to it and cause a clumping of the red blood cells. And so the effect is that when you have a lot of virus present and you put the red blood cell mixture, virus mixture in a round bottom well, these are very small wells. If there's no virus present, the red blood cells will tumble to the bottom of the well and make a little button. But if there's virus present, the cells will make a meshwork along the walls, and you make dilutions of virus, as you can see on top here. And then finally, when you get out to a certain dilution, there's no more aggregation, and you get the little buttons again. So you say, oh, the HA titer is 1 to 512 for this sample. So you'll see a lot of HA titers in the literature. Hemagglutinin inhibition, or HAI, we do this assay, except we have antibodies to the viral HA, and we want to know if they will prevent hemagglutination. And that's an early indication. That's how we learn early on whether the flu vaccines are working against circulating strains. We can use immunological techniques to detect viral antigens or antibodies. That's called ELISA. So in one version, if you want to look for viral antigens, you have an antibody to the antigen, which you put on a plastic dish, and then you put the sample with your antigen on. If there is antigen in the sample, the antibody will bind it, and then you can detect that antigen with a second antibody that has some kind of indicator that you can easily measure, like fluorescence. You can also measure antibodies against viral proteins. In this case, you put the viral protein on a plastic support. You now have a clinical sample that you think has antibodies in it, nasal wash, serum. If there are antibodies to the viral protein present, they will bind to the antigen. Then you can detect the antibodies with a second antibody that also has an indicator on it. So here is a real world example of immunofluorescence, which is what we're talking about. Using those cells that we talked about at the beginning of this mini lecture that were infected with SARS-CoV-2 from the bronchoalveolar wash of that patient, that ICU patient. They used an antibody against the viral nucleoprotein of SARS-1, which cross-reacts, now we know, with the SARS-CoV-2 nucleoprotein. And these infected cells you see are lighting up by immunofluorescence because they're infected and they contain nucleoprotein. So you add, you permeabilize the cells with a reagent. You add the antibody, it gets into cells, it will bind to nucleoprotein, and then use a second antibody with a fluorescent tag attached to it to see it. Now, here's a version of the rapid antigen test that was eventually developed for SARS-CoV-2, in which many of you have used and will continue to use. Here we take advantage again of the use of antibodies. We have a sample pad, right? This is the plastic pad that you can buy as part of your COVID test, and has this pad is showing in a tiny window. So I've expanded it here for clarity. So it has 3 regions on the pad. So the pad refers to an absorbent material that if you put a drop of fluid at one end, it will wick to the other end. So the fluid will move through it. So it's called a wicking pad. So in this first area, we have antibodies to the antigen you're looking for. In the case of COVID, it would be nucleoprotein, right? Linked to colloidal gold. So these are embedded into the pad. And then downstream, you have monoclonals attached, which are directed to the antigen. And then the last one is a control line where you have antibodies against these antibodies right here. So you put a drop of nasal wash here. It wicks down the pad. If there's antigen present, it will bind these antibodies and they will continue to flow by capillary action. So now we have the antigen in red bound to the antibodies. Then these antigens will bind the antibodies in that test line. So now we have the viral antigen binding the antibodies on the wicking pad, and those antigens are also linked to the original antibody that was in the pad. And these are linked to colloidal gold, those yellow circles, and the gold makes the dark line. So this tells you that you have antigen, that's your positive test. And then some of the antibody antigen complexes will keep flowing to your control line, and the antibodies will be picked up by binding to antibodies embedded in the pad, and those also have colloidal gold. So this will show up as a black line as well. So 2 black lines means you're positive for nucleoprotein. 1 black line at the control means the assay worked, and if you don't have a black line at the test, you're negative. But if you have 1 black line at the test and nothing at the control, the assay didn't work, and you should just do it again. So that's how these rapid antigen tests work using antibody-antigen interactions. And finally, a few nucleic acid methodologies. Of course, sequencing has come to the point now where we can take an isolate from any patient and do the whole genome sequence. It's based on the fact that you can do many sequences at once in a short time. And that, of course, is used to identify viruses, to identify new pathogens. And the human genome can now be done for $1,000 in one day using these deep, high-throughput sequencing methods. In the early days of the human genome, it took 10 years and $3 billion to do that, the first genome. Now it's $1,000 and it takes one day. By the way, this is a DNA sequencing gel of the kind I used to run. This is not deep, high-throughput sequencing. These would take a long time to get a genome sequence. And I did the genome sequence of poliovirus, 7,440 bases, using this gel methodology. It took a year to do that. Anyway, when you have a sequence, then you can build a phylogenetic tree. So here we have 10 isolates of a particular virus, and we take their genome sequences and we run them into a computer program which says, put together the ones that are most related by genome sequence. And so here you can see viruses 1 and 2 are more closely related than any of the other pairs. Same with 3 or 4. Less so with 5 or 6. They don't pair with anyone, 5, 6, 7, 8, but 9 and 10 pair. And then you can tell how these viruses are related to each other. For example, viruses 1, 2, 3, and 4 have a common ancestor shown in green. Virus 1 and 2 have a common answer. 1, 2, 3, 4 ancestor. 1, 2, 3, 4 have a common ancestor. 1, 2, 3, 4, 5, etc. You can keep building back these common ancestors until you get to the root of the tree, which is the presumed ancestor of everyone. Now, the blue and the green squares, we often don't have those viruses. They're inferred from the genome sequence. But you can now say, I have virus 1 and virus 10. They're not so closely related, but I have 1 and 2 are really closely related. So if 1 and 2 are in 2 separate patients, they were probably infected by a person with a common ancestor. So these trees measure the genetic distance between their organisms. They identify the nearest relatives. And this x-axis here, by the way, is the degree of genetic change. And the numbers that are given here are the number of changes per length of sequence. And this was done, of course, early on in the COVID pandemic. And what they did is to get the genome sequence of that novel coronavirus that they found from the patient. And this was in the same paper that we were looking at before. And did a phylogenetic tree with all the other known, or many of the other known coronaviruses. So here in red are the genome sequences of the new coronavirus, which we now know as SARS-CoV-2. The closest ancestor was the bat coronavirus, RATG13. But again, a few percent different, 1,200 bases. This was not the ancestor of SARS-CoV-2. But the important point here is up at the top. These are here is SARS-CoV from 2003, 2 different isolates, very distant from SARS-CoV-2. So it's clearly not SARS-1. It was a very different virus. And these are all different bat SARS-related coronaviruses that have different relatedness to SARS-CoV-2. And down here is a human coronavirus 229E. So this is the method that you use to look at the relatedness of viruses. And finally, you can use PCR, of course, to detect viruses. This was done early in the pandemic. The PCR testing was replaced eventually by rapid antigen tests. PCR is a methodology that's widely used and depends on amplification of small amounts of nucleic acid. Let's say you start with a small amount of DNA in a sample, or if it's RNA, you convert it to DNA using reverse transcriptase. You can have vanishingly small amounts. You subject the DNA to heat to separate the 2 strands, and then you add primers, DNA primers, which are shown in red, and then a DNA polymerase, which will use the primers to copy the strand of DNA. And so you go basically from 2 strands of DNA to 4. And then you repeat this, and you have 8 and 16 and 32. So you can see you have logarithmic growth eventually of the number of chains, and you can detect a very small amount of starting DNA or RNA. But these reactions are all done in the same tube, heating to high temperatures and cooling. They require an enzyme, a DNA polymerase that's thermostable. And that was discovered in the 1960s by a microbiologist, Tom Brock, who was studying the hot springs in places like Yellowstone. and found bacteria growing in them. And it turns out that, of course, they grow at high temperatures, that the DNA polymerase of these bacteria can withstand the high temperatures of PCR. And that's how Kary Mullis and others developed PCR and eventually got the Nobel Prize for that. Fast forward, you can use PCR to detect SARS-CoV-2. But I want to end up with a warning. DNA product detected by PCR is not the same as infectious virus because you are looking for very short segments of DNA between these primers. You don't amplify the whole genome. Here's an experiment done with Zika virus, which shows what I'm talking about. If you infect male mice with Zika virus, the virus is shed in seminal fluids. Here's the time of shedding. Infectious virus is shown in red. And so infectious virus is shed for about 21 days. But if you do PCR on the seminal fluid, you can detect viral RNA for up to 60 days. Yet there's no infectious virus there. They're just pieces of RNA that hang around. And so early in the COVID pandemic, we had people being PCR positive for weeks, and they would say, "No, you have to stay home," without figuring out if there was infectious virus there because they didn't do infectivity assays. If you vasectomize the mice, very little infectious virus in the seminal fluid, but still Zika viral RNA. So PCR product is not the same as infectious virus. And this was found eventually for SARS-CoV-2. Here's a number of samples from patients at different times after infection, days 2 to 22. The green is the quantity of RNA detected by PCR. So you can see you can get PCR positivity for up to 20 days, yet the infectious virus is gone at day 9 or 10. And so here's the plaque assay of SARS-CoV-2 on the right. It can be done. It's just that the virus is a BSL-3 pathogen, and it's really hard to have that in many different places. So, we should have known this, and we needlessly quarantined people for many more days than they had to be because we didn't realize that RNA viruses, when they infect you, the infectious viruses are cleared eventually, but they're pieces of RNA that stay on for a long time. Okay, that's all I have to say for today. Let's turn off all of this screen share, come back to me and you. Let's see if you have any questions. In the early COVID days, it was driving me crazy that many people assumed that if you were PCR positive, you were infectious. No, that's because they didn't know virology. And of course, we would talk about it, but no, very few people listen to us. So let's see what we have here. I have to scroll down. Here we go. John says, Shane Karate published in Nature, Immunological Memory, Diversity in the Human Upper Airway. Hey, we could get Shane on TWIV. He would love to come on, right? It's a good idea. Let me write it down. Now we could also ask him a lot of other questions, right? Thank you for that suggestion. Brian says, we have a family member who got a first Shingrix dose last November. Never got the second dose last week, contracted what is likely shingles. So we got this question at Daniels, right? And Daniels said, wait till the infection is resolved, however long it takes, and then get the second dose. These are Daniels' recommendations. I am passing them on to you. I am not giving medical advice, okay? Thank you, Rob, for your support of science communication. Appreciate it very much. Rach from the Isle of Man says, so many people here believe COVID was a lab leak. I tell them to watch TWIV, but they don't. It's very frustrating. Yes, it's very frustrating. I don't know what to do about it. I mean, we've done our part. We could certainly do more, but there's so much else to talk about. Only when it has consequences will we step in. Right now, well, there are actually consequences, but you don't know about them, and I can't tell you about them. But saying it came from a lab has consequences. Yeah, I'm sorry they don't listen to TWIV. There's a 50-minute version. Is that too long? Come on. Sports are longer than that. They don't want to listen. They're not interested. Actually, I have to make a 5-minute video explaining why it didn't come from a lab, right? I have wanted to do that for some time. Just write it out, record it. Here's why, and make it interesting so that a lot of people will see it. I'll try that. Artemis said early 1980s, Geneva University didn't have any specific virology courses. We learned about phages and tobacco mosaic virus in the genetics and biochemistry course. Yes, that's how you learn most virology in other courses. But I would argue that you should have a virology course. First, I'm biased, right? Because I think they're amazing viruses. But, I mean, look, we have a pandemic, but we also have outbreaks every year. And people should really understand viruses. They should understand that PCR positive is not infectious, right? Everybody should understand that. So the more undergrads that are taught, the better. My lectures, my virology lectures are on YouTube. They're free. Any professor teaching a virology course can use them. I get many requests from people to do that. It's okay. Use them to help you teach your course. The point is, not everyone is a virologist, right? I'm a virologist. I've spent 40 years working on viruses. I can impart that information to you, and then you can use it to teach a course. Yeah, we have 120 viewers and 92 likes. Please hit the like button. It's really easy. It's right below your screen. Just hit it. John wants to know, will those who received chicken pox vaccine and never got the disease need the Shingrix? Yes. Yes. Because you may have had a asymptomatic infection and you actually do have chickenpox virus genomes, VZV genomes in you. So you should get Shingrix for sure. All right, here's some weather information from Patricia. The Atlantic hurricane season started in June and ends in November. Hurricane Beryl, Tropical Storm Chris. The next name is Debbie. Okay. Speaking of the weather, I got an email from a very nice man the other day who said, your TWIV is, I love TWIV, but it's too long. You got to cut out the weather. And I'm like, I didn't answer, but what is the weather? 5-6 minutes? Wouldn't do anything to cut out the weather. There are other things if you want to really make it short. I mean, we could do one paper and no pics. Then it would be 30 minutes. But that's not what TWIV is. It's about chattering, right? And to listen to how scientists think. So I'm sorry it's long, but anyway, over the years, a few people want the weather gone, but most don't seem to mind, and many like it. So it's not going anywhere for now. Elise says, I heart science. I wish we all loved it because then we would appreciate it. and not just us, the lovers. Yeah. Okay, M asks me, outside of the sciences, what discipline subjects tend to inspire your work or research? Well, I don't do any science research anymore, right? But I think literature is a big influence. You read stories, especially about, it could be fiction or nonfiction, about research, like Aerosmith, right? Which I have in the incubator. Aerosmith. It's very inspiring. Um, I think because I did research in a non-media era, right? No, very little videos and podcasts only later in my career. Because now I could see that you could get inspired by videos, right? You go watch a YouTube video, go, wow, I want to use that technique. But that wasn't me. It was mainly literature. And that's not surprising because my mom was an English teacher and we read classics in her class. Well, I wasn't in her class, but I used to read them anyway. So I think it was for me literature. It's the big one. It's a good question. So more weather from Margot. Right now, the humidity is 30% in Phoenix. It's pretty dry. Quickly increasing. It was 46% a few days ago. Monsoon rain still skipped us. Tucson has been getting all the monsoon this year. And Marco said, that's humid for Phoenix. Much has changed since the 70s. Ian says, Dunedin is Gaelic for Edinburgh. Ah, yeah, I always pronounce it wrong. Thank you. I say Dunedin, right? Schmuck. Me. Dunedin. I'm going to forget that. Dunedin. Hey, I'm Dunedin. I write it down, but I'll never see it. Thank you. Dunedin. Dunedin. Stress on the dun. So it's not Dunedin. It's Dunedin. Dunedin. Yeah, Dunedin. Okay. Dunedin. Right? Sorry, folks, to put you through that. Artemis says there seems to be a COVID wave in Quebec. Yep. There does. Many places. Here in New York. Yeah, here Mike says summer peak etiology, planes, trains, and autos. Yeah, people getting right next to each other, for sure. Yep. Environmental Coffee House writes, David Feldman, my Emmy-winning comedy writer, Seinfeld, and political commentator, has contracted COVID for the first time. I wrote him to advise him to watch this channel and learn. You should particularly listen to Daniel's clinical update, right? That's fast. At least you get the story. Thank you, John, for your contribution to science communication. John made his donation to this link on the screen. These programs are invaluable to have experts in their fields select and discuss papers and research to a layman like me. So that's TWIV and TWIM and TWIP, etc. So office hours, this thing is a way of having you ask questions live. It's not always just me, of course. I often have guests, and I'd like to have more guests. It's just an odd hour, right? For the East Coast anyway. But the idea was that you could ask questions of people live, which I think is good. So thank you, John. We appreciate it. SM writes, my wife and daughter got COVID. My wife's fever went away 3 days before meeting up with family for vacation. My daughter stopped having a fever 1 day before we went. And then this continues. My grandma got sick 2 days after we got there, but no one else until 3 days after. And 6, my grandma was always furthest away from us. Why did no one else get sick first that held and kissed my daughter, but my grandma did? Okay. um, because everybody is different. You can have a family of 6 people, an infected person comes in, nobody gets infected, one person, or all of them get infected. Because everybody has a different, they're all different age, they have different immune responses, and they have different exposure histories as well now to this virus, different vaccination histories, different responses. So you can never say that just if you introduce an infected person into a family that everyone's going to get sick. No, that doesn't hold. Not at all. And so I don't know why specifically only the grandma got infected. I would guess that the person was shedding not a lot of virus. The younger people were great immune systems, able to hold it off. Grandma's older, weaker immune system. It got her. That's my speculation. Laura writes, Rainbow Lake in Connecticut, the storm just passed and maybe there's a rainbow over the lake. Humid, of course. It's extra humid near the lake this time of year. Cool. You know, people don't like weather, but people like to talk about it. Can't do anything about it except like have an umbrella or a raincoat. Oh, here we go. SM had another. Does it make more sense that my grandma got COVID from someone else when we were out on the boardwalk? Or that's hard to know, right? Were you close? I mean, you were closest to your daughter. She was known to be infected. I think the most likely explanation is that grandma's more vulnerable. But I agree. You cannot rule out that she got it outside of the house. In fact, the only way you could is to sequence the isolates from everybody, and then you could figure that out. But obviously, that's not been done. So I would say the most likely is that she got it from the daughter, but it's also possible outside the house. Yeah. All right. Ian says from Dunedin. A while ago, I asked why we don't treat bacteria with multiple antibiotics concurrently to preclude resistance. Why wouldn't it work? Is it due to a property of bacteria? We should have asked Michael Schmidt when he was here, right? I don't know. It's a good question. Should we get Michael Schmidt back? Yeah, here's an article in Science. Combining antibiotics may backfire, far from knocking out an infection. Multiple drugs may make it more aggressive. You might think that combining 2 antibiotics would be a great strategy. A new study suggests that a 2-pronged attack can backfire badly by giving super-resistant bacteria the opportunity they need to come out on top in the struggle for resources. So this is a team from the University of Exeter. They put this synergy idea to the test. They did an experiment with 2 antibiotics, and they found the first day a 95% reduction in bacterial growth with 2 antibiotics, and then a population explosion, which they didn't see in singly treated bacteria. So that may be one answer, that it encourages resistance, which is counterintuitive. But the idea would be it's now selecting for the rare bacteria that are resistant to both. This is different from what happens in viruses, right? Brian wants to know if I have any thoughts about why COVID has seriously affected old people so much more than young people. I think it's all about interferon. And the big one is having antibodies to interferon, autoantibodies, right? So it turns out that about 20% of the population have these autoantibodies. They track with having severe COVID. And if you look at the age distribution in the population in the US, the number of people with autoantibodies to interferon increases with increasing age. So I think a big part of it is that older people have antibodies to interferon. But it's only one part. I think that's an obvious part that we teased out, but I think, being exposed to a new virus in your 70s, you have trouble making memory B and T cells. And so you may survive that first infection, but you don't have good memory cells and you won't survive a second one. But I think the interferon is a big one for sure. Hello, Andrew. Andrew is here. Our moderator got delayed by flooding. Better late than never. Ian says, it's cold down here in Otipoti. My solar panels haven't been producing electricity as it's cloudy. So here's the question I referred to before. The incubator in the lab, will you have a lab in the incubator? I think not. Although, it'd be cool if we had an extra room and you could put a lab in it, a simple lab that didn't have any dangerous experiments, a BSL-1. But I don't know. We have a lot of stuff to do right now. That could be a future thing. Visto asks if HeLa cells could be infectious to people histocompatible with Henrietta. Oh, they did a horrible experiment about that. Let me look up the details. Yes. Before the Nuremberg Code was established after World War II, Chester Southern injected HeLa cells into humans without their knowledge that the injections contained cancer cells, before the era of informed consent. I think in a few cases they got little local growths, but nothing serious, fortunately. But histocompatible with Henrietta. I don't know if that would be enough, because the tumors, the cells have neoantigens that are unique to Henrietta, right? And so they may be rejected in a healthy person. I don't know. I didn't see EG before from Mississauga, Ontario, Canada, 31C and hot. Yuxing asks, is this a live stream? Isn't it the virology lecture? Yeah, this is a live stream, and I make mini lectures. Because why? Well, first of all, not everyone has seen the lectures, the virology lectures. Second, it never hurts to repeat. And third, you can ask me questions, and I can present it in a slightly simpler way so that you understand it, right? For non-virologists. That's the idea. Kang wants to know, is a cluster of cytopathic effects count as a plaque? Sure. Cell killing is a cytopathic effect. And so when the cells are near each other, they make a plaque. And the reason they're near each other is because we have an agar overlay that restricts diffusion of the virus. So the virus that comes out of that first cell infects the neighboring cells and the neighboring cells and the neighboring cells, and the plaque gets bigger and bigger. But yes, it is a collection of cytopathic effects. Does the immune system work 24/7? Yes, it doesn't rest. Those cells are patrolling you all the time because pathogens don't rest, right? And we have normal flora in us that have to be monitored all the time, right? So yes, 24/7, those cells are flowing around, those sentinels in particular. Macrophages, dendritic cells are patrolling you all over the place. You have local and you have resident T cells, you have systemic T. They're all 24/7. Absolutely. Are there any common viruses, bacteria, and parasites that infect the brain? Yeah, there are many, right? Common. Well, poliovirus infects the brain, right? But it's not common anymore. But there are many other viruses that are in our brains and bacteria. I mean, if you sequence a brain, you can find viral and bacterial sequences. I don't know if they're infectious virus. I don't know if they're living bacteria, but they are there. Yeah, parasites, sure. Toxoplasma and neurocysticercosis. Listen to TWIP. You'll hear all about them. John said 1 hit is linear, but I'm still trying to wrap my mind around 2 hit being parabolic, second degree. Okay, so I'll read to you from the slide, right? Which is, let me pull it up here. Here we go. For 2-hit kinetics, the number of plaques is directly proportional to the square of the concentration of the virus inoculated. That's why you have the parabola, okay? Yuxing says, I know there's a CCID50. What's the difference between TCID and CCID? It's the same because in the example I showed, each of those little wells is a cell culture. So it could be cell culture, infectious dose, tissue culture, infectious dose. I prefer cell culture actually, but I don't like tissue culture because they're not all made from tissues, right? Then there's paralytic dose, 50%. There's the lethal dose, 50% if you're working in animals, right? So okay. Laura says HeLa is a very cervical cancer causing form of HPV. Type 18 is what Henrietta had, not 66. Yes, so she died of cervical cancer. And if you sequence the genome of HeLa cells, you can see the oncogenes of HPV 18 integrated into her genome. Did you mention what is happening during the eclipse period? I didn't. So thank you for asking. So during the eclipse period of an infection, you infect a cell with a virus. There's a period where nothing happens. There's no apparent infectious virus produced. I shouldn't say nothing happens because lots of stuff is happening. You're making the parts to make new viruses, right? You're making genomes, whether they are RNA or DNA, and you're making proteins, and they're assembling into new virus particles. So that takes time. And that's why there's a period called the eclipse where you don't see new infectious particles. Yeah. Artemis says, is there a fast lab test for detecting shingles? Yes, there's a PCR test, and there's also an antibody test for detecting shingles. Yep. There, Max says, too hot to sleep, so time for some virology. Well, good. I'm glad. Sorry you can't sleep, but welcome. Yes, Barb Mack, our other moderator. Welcome. John asks, which reproduce more quickly, the fastest viruses or bacteria? Well, so some phages can reproduce in 20 minutes, and so can some bacteria. Let's look. So the fastest doubling bacteria, Vibrio nitrogens, can double every 10 minutes. E. coli does 20 minutes. So let's now look for the fastest doubling virus. And now the responses are muddled. It's got to be a phage. So let's look for phage. Yeah, we're looking at lysis at 20 minutes. Wait, since the fastest doubling time for E. coli is about 20 minutes. Well, I would say that this bacterium of 10 minutes has it, but there could be some faster phage. I think it's pretty close between phages and bacteria. Patricia, nice hexagon. Summer Full Metrics 2024 July. Which variant is directly responsible for the rising numbers? I believe it is what is it? JN1. I was just looking at this earlier today. LB1, sorry. JN was an earlier one. So remember, there were flirt variants earlier in the summer, and then they led to LB1. And the flirt subvariants were most of the cases at the beginning of July. KP3 was one of them, KP2. So KP3, 37% of cases in the US, KP2, 24%, KP11, 9%. And then LB1 is similar to the flirt strains, but has an additional change. Was responsible for 15% at the beginning of the summer. So I guess that LB1 is responsible for the latest surge. Are the wicking pads made in Iceland? I don't know what a wicking pad is. Why Iceland? There must be some other thing in Iceland that's called the wicking pad. Let us in on the joke, Andrew. Tony says, I've tried doing validation studies for 2 different companies. Rapid COVID tests and expected results were horrible. We could not even validate. And my hospital lab had to stick with PCR. Well, there were clinical trials done, and they had no problem validating totally. So I don't know what you're finding. Yixing says, revisit the basics of virology to recapture that familiar feeling. Yes. Now you know how rapid antigen test works, right? It's great to learn how these rats work. John says, is it that false positives are much more rare than false negatives? Yes, that's correct. False negatives, the assay is not working, or there's the antigen. So the antigen also wanes, I think. And also, when you think about sampling, the nasal mucosa is not going to be uniformly infected. So you may hit a patch where it's not infected one day, and another day you hit a patch that's infected. It's not perfect. It's not like taking blood. We have a nice sampling of everything that's mixing in that blood, right? Nasal swab is a little iffy. Les asks, how much of coming up with a phylogenetic tree is human interpretation, or is there only one possible tree? No. What you do is you can adjust the parameters of the algorithm that the computer is using. and you can set different cutoffs for relatedness and so forth. You can generate very different trees from the same sequences. And that's one of the problems with bioinformatics, right? Is that if you tinker with the algorithm, you can change the outcome. So you have to be careful. In the paper, they tell you how they make the tree. I mean, there's some standard programs that people just plug and play, right? And they don't change anything. But if you're doing your own, you can change things. Yeah. Hmm. John remembers the CT cycle threshold, right? Cycle threshold. Oh, there's a little spider here falling right in front of me. Can you see it? Here. Oh, you can't see it. Maybe there it is, right there. See? Well, I don't want to kill it. I'm going to put it on the desk here and let it find its way. Cute little spider. The itsy bitsy spider went up the water spout. CT cycle thresholds. So you're running your PCR each cycle. And this is PCR that's done when a dye is incorporated into the DNA, and you read the intensity of the dye. And the more DNA you get, the more dye, the more intensity, until it goes above background. And that's the cycle. And you count how many cycles it took to get to that. So if it takes 10 to hit the cycle threshold, there's a lot of DNA there. But if it takes 40, there's not a lot of DNA. And that's what we should have been looking at early in the pandemic. You know, if there's a CT of 40, there's not a lot of DNA there, even though it's positive, right? Now, the other issue is that every swab is going to have a different amount of DNA, right? Because it's not standard. So you have to be able to standardize to it, to something internal. And I would say, what would be a good standard? Mitochondrial DNA or ribosomal RNA? Just to say, okay, we're going to normalize to the amount of total nucleic acid that we have here, but that was not done. And so people were not very confident in using cycle threshold as a basis for saying you don't have to quarantine, or you do. Lessons learned, right? Visto says, oh my God, DNA polymerase in volcanic water. Life finds a way. Yeah, these bacteria that live in the hot springs, they have to have thermostable everything. And Tom Brock, used to study this. And then these guys were thinking of thermocycling, and they say, wait, there are these bacteria that grow in the hot springs. Maybe that would work. And they purified the polymerase from it. The bacteria was called Thermus aquaticus, and the polymerase was called TAQ polymerase. It's the first one. There are others now, but it was the first thermostable one. Today we have fancy thermocyclers where you mix your reaction in a tiny tube, you put it in, you push a button, and you go home. The next day it's done 40 cycles. But in the old days, we had to do it by hand. You'd have a water bath at one temp and a water bath at another temp. You'd put it in one for the amount of time, then you move it to the other, and you move it to the other, and you just do that 40 times. You had to stand there and do it by hand. Wow. All right, Artemis stepdaughter was very sick, gastro symptoms, extreme fatigue, COVID home test negative. If it was COVID but without respiratory symptoms, would you expect the nasal sample to contain SARS-CoV-2? Yes, because that's how it got in. It's not going to come in through your stomach. Not happening. It's coming in through your respiratory tract. And the gastro symptoms are probably inflammatory based cytokines, systemic cytokines. So yes, should be positive PCR. Of course, depending how long after, if it's more than 5-7 days, it could be an inflammatory response where the virus is no longer present. Yeah, Tony remembers how long those old cyclers took. Now ERMDs complain when their PCR takes over an hour. It used to take 16 to 20 hours. Yep, you got it, Tony. So now I know Tony is real because he knows how long it took for cyclers. Is SARS-CoV-2 still BSL-3 today? Well, the WHO has said that most experiments can be done under BSL-2, but CDC in the US has not said that. So it's still BSL-3 in the US. You're welcome, Visto. I'm glad you liked it. These are principles that apply to all viruses. I thought it would be important. Can prions be transmitted, spread through saliva? I know it can in deer, but in people. Yeah, in some animals, in deer and sheep, but not, it doesn't say in people. So in deer and sheep, the prions accumulate in the salivary glands and therefore can be spread through saliva, but I don't think that happens in people. Yes, Shane Crotty is a TWIV regular guest star. He'd be happy to come back, I'm sure. Ariozone likes a 5-minute video on why SARS-CoV-2 did not come from a lab. I will make it. I will make it interesting, and hopefully you spread the word. Suzette liked the mini lecture. You're welcome. I'm glad you liked it. Lisa, do not cut out the weather. We won't. The local weather is irrelevant to a worldwide audience. It is. That was one of the points of the letter writer, and also said by the time it's released, the weather has changed, right? Which is true. But I think it's more than the weather. It's us warming up. We're just chatting. And it's only 5 minutes. I don't want to talk about politics. I don't want to talk about something else. It just happened organically, you know. The weather is the icebreaker that gets people talking, as if we need an icebreaker, right? Because we know each other for so many years, but it's just stuck. Yeah, WHO has reclassified it, but the US has not. And it's not all experiments either. Yeah, the WHO downgraded it, but as far as I remember, CDC did not, and they're the bosses. And yes, if you listen to the podcast, you can fast forward to the parts you don't like. I don't see why that isn't the solution, right? Yeah, Artem is weather on Twitter. If someone doesn't like it, forward until you're past it. That's exactly right. What the heck? Rabies virus migrates 12 to 24 millimeters a day across the CNS, right from the peripheral site of inoculation, gets into the neuromuscular junction, then it's moved by axonal transport, which is relatively slow, and then moving within the CNS. Also, both by fluid flow and axonal transport, and some reproduction as well, but it's mainly axonal transport, yes. Les wants to know what percentage of sarbecovirus variants are found in nature versus a lab. If we don't secure live animal markets, we risk future pandemics. Well, there's a huge 99.9% are in nature. You know, labs have only sampled a vanishingly small number of variants, right? You're right. I mean, there are many kinds of live animal markets throughout the world, but the fact is that this part of the world, in China, in I would say Asia in general, it's not just China, but Southeast Asia, have the bats that harbor the viruses that have now multiple times spilled over into people, twice through live animal markets, right? So it would seem prudent to secure them. I understand it's a cultural thing, but you could do things to mitigate the danger, right? It would cost money. And that's the issue, right? It would cost money. So you could check every animal that came in. It would cost money. And profit is always the driver of everything in the world. But I agree. Yu Xing, could you review the development history of the RSV vaccine briefly and give us a general overview? Why don't we make that a mini lecture next time? Mini lecture, RSV vax history. It's a great history because the first attempt was made in the 1960s and it failed. Kids got sicker who were vaccinated, and that put a chill on vaccine development until recently. And now we have a couple of vaccines and some monoclonals. But yeah, that's an interesting history. I'll do that next time, I promise. I know you asked me that last time. Is rabies a virus? No, of course it's a virus. It's not a prion. There are prions, but rabies is a bona fide virus. It's a rhabdovirus, negative strand virus. Yeah. So Yuxing wants to know, why did the vaccine start decades ago but only made significant progress? So basically, they made a vaccine in the 60s. Kids died after they got infected who were vaccinated, and nobody would touch an RSV vaccine for 50 years until they figured out what went wrong. It took a long time. And I'll tell you about that next week. Yes. Yes, rabies has the world's worst kill rate of all the viruses, 99.9% fatal if you're not vaccinated. Yep. Ian says, I was wrong. Stress as you did on the E. Right. So it's Dunedin. Dunedin. Thank you. Laura read about SARS-CoV-2 in backyard animals, opossum, fox, mouse. Yeah, it's infecting many, many different animals. Artem is appreciating the opportunity to ask questions from experts. Thank you. That's the whole idea. Less with sexual reproduction, you get a chance to introduce different MHC genes so your kids can ward off more rounds. That's the whole idea of sex, to fight off parasites. Yep. You like Michael Schmidt. Yeah, I'll bring him back in time. Yes, VSV oncolytic therapy. You know, it doesn't figure in my oncotherapy lecture. I'll look into it to see if there's anything in trials. VSV oncolytics. It's a good question. Can you correlate COVID-19 with low mucosal respiratory temperature? We just had a letter on Twiv about this. Let me look it up. Yes, Susan Weiss wrote in. Okay, let's go to the letters. Yeah, this is going to answer your question. Listen, common cold coronas replicate better at lower temperatures, 33 versus 37. This is due to increased innate immune responses at higher temperatures. This may contribute to why the cold viruses replicate mostly in the upper tract and do not progress into the lung. In primary nasal cells, SARS-CoV-2 replicates equally at 33 and 37, but at late times post-infection, when innate responses kick in, it replicates better at 33. MERS replicates in both and therefore can go into the lower tract. So maybe later times in infection, it does correlate. No, at later times, it replicates better at 33, which means it shouldn't be in the lung. So I think the jury is out. We don't know yet. Viv is teaching so much with COVID antibodies. Is there any theory where someone is low dose exposed often, this boosts the antibodies? Yes. I mean, in fact, Shane was an early proponent. Shane Crotty was an early proponent of that idea. But the problem is that with the emergence of variants, so whatever you're first exposed to is what you're mainly going to make a memory response to. And if you get boosted with variants, they may preferentially induce antibodies to the original, and that may not help you so much. It's not clear. So Shane probably can shed some light on this. We'll ask him. Rainey says one would think that the experts at CDC would have been knowledgeable of the fact that a positive COVID PCR was not indicative of contagion. Any idea why they mismessaged on this? Now, I think that a lot of epidemiologists at CDC who don't know the fundamentals of virus biology. The epidemiologists are running the polio eradication campaign, and it's not working because they don't understand basic polio biology. So maybe that's part of the problem. If you don't read the literature, you're not going to know that for Zika. Well, actually, that was a CDC paper. So they should have known that Zika paper figure I showed you, that came out of the CDC. So there were scientists there that knew that. I don't know why they mismessaged. I think they felt they couldn't rely on cycle threshold because the sample sizes were different. But I don't really know. You'd have to get someone from there to ask them. Did interferon antibody mitigate interferon-induced fever? In theory, it would, right? But I don't know what happened there. I think you should call the kitchenette the lab. Yeah, we have a kitchenette at the incubator. Okay, let's see. We should wrap up here. A lot of good questions, but we are going to have to. Davey says someone has not caught COVID, but opening up to the world. Is there a benefit catching it straight after a booster rather than 8 months? I don't think there's ever a benefit because everybody's different. Maybe the vaccine didn't work. You don't want to chance that, right? I think you need to, give the vaccine a couple of weeks to work, and then you can go about life. But I wouldn't try and catch it on purpose, right? You may get it, and then hopefully you're protected by the vaccine. But I don't think you should try and catch it. All right. When sequencing, how do you know sequencing all the bacteria and other junk that is ever? You do. Whenever you do a environmental sample or even a clinical sample, you get sequences from a lot of stuff. You have to computationally remove all that, subtract them computationally, as we say, and then what you're left is your virus sequence. Last week on TWIVO, we talked about the genome sequence of a woolly mammoth. They had skin and they extracted DNA. Only 2% of the sequences were woolly mammoth. The rest was bacteria and other cells and crap in it. And they had to subtract it out. Yeah. So you don't isolate what you want to sequence. You sequence the whole thing and then you can computationally remove everything else. Okay. Oh, here we go. Wicking is the German way of saying Viking, hence the Iceland pun. I got it. Oh, this is an interesting. The Seine, right, where the swimmers or some of the swimmers are going, like the triathletes, was contaminated the other day with fecal coliforms. And no, it's not a good idea. But the swimmers say, yeah, we do it all the time. It's not a good idea. You're going to get bad infections in all different places, right? Not a good idea. All right, it's 10 o'clock. One more. Is there any news on why shingles leaves the nervous system? We don't. We know why herpes simplex leaves the nervous system, but we don't know the stresses that cause shingles to leave. No. Oh my gosh, so many cool questions, but it would take me another half hour to get to them. So bring them back next week. Okay. Let me thank everybody for being here. Our moderators tonight were Les, Barb Mac, UK, Steph SF, Andrew. I think I got everybody. We didn't see Tom tonight. Thank you for moderating. Really appreciate it. And thank you all for coming tonight and having great questions and way more than we got to. And they're all good. So please bring them back next week. Okay. I really encourage you to. That's August 7th. Okay. I'm going to copy my office hours and put it on to August 7th, 8 p.m. Eastern. A lot of leftover questions. We will come to them next week. I really like them. Thanks to moderators. Thanks to you for coming. We had 135 people here at one point. Thanks for your questions. And I think thank you all. We'll see you next week. In the meantime, please take care of yourself and be safe. Where's my plaque assay thing? I can never find it. Here it is. Good night, everybody.